rightly said, if you know you are on right track, if you have this inner knowledge, then nobody can turn you off no matter what they say. Whenever there are no ceiling, the sky's limit. So let's keep until every one of the million women and girls across the world has the opportunity she deserves to have. A very pleasant morning to everyone present over here. Our today's esteemed guest speaker is one of such well-known Indian scientists who has paved a million of Indian girls and women. She is an inspiration of thousands of youth of our nation. Her constant research and development in the area of bioscience is example of strong determination and willpower to get succeed in the life. With this note, I, Dr. Dwani Upadhyay, Assistant Professor, Microbiology Department, feeling honored to welcome our eminent speaker, renowned Indian scientist, Professor Dr. Sandhya Shrikant Vishweshwarai, on behalf of Parul University. Ma'am, we are truly fortunate to have you with us today. I feel very delighted to welcome our HOD, Professor Girish Babu Sir, Department of Life Science, Parul
Good
University.
morning.
I would also like to welcome all the faculties and my dear students to this one kind of live webinar. I assure you that experiencing this live webinar will uplift your knowledge with lots of learning and lessons. I would like to humbly request to our HOD, Dr. Girish Babu Kesar, Department of Life Science, to say a few words on this august occasion. Please, sir.
Thank you, Dhwani. Good morning, all. It is again one more fantastic opportunity for all of us that we are getting an very good opportunity to listen to Professor Sandhya ma'am here. So, especially the Parul University is having such a platform where we, all the eminent people from India or, even from outside the countries, we get an opportunity through this platform where the eminent people would be sharing their experiences. They help us to understand the concepts and the updates of the science, and even they guide us, the young minds who are there, who are looking for all these kinds of the guidance for their career. It is really an immense pleasure for us to host you, Sandhya ma'am, to talk to our people. I mean, like the students, the faculty, and other fellows who are working in the Parul University, not only in Parul University, even from outside the Parul University also. So I take this opportunity to welcome you formally to have this webinar. We are eagerly looking, and we are eagerly looking for these kinds of the updates what you are going to share with us, and the guidance you will be giving to the young minds who are there. And we are really happy to have you here. Welcome you on behalf of Parle University, ma'am. I request you to I request you to share your experiences. Thank you so much, ma'am.
Thank you. Thank you.
Thank you so much, sir, for your boosting words. You always enlighten us with your words. Our eminent speaker is one of such personality who did not choose to keep herself confined, but had decided to sail her ship made with encyclopedia of knowledge and years of research work for putting up extraordinary efforts in creating the pathway for the women of India in this science. Although Honorable Professor Vishweshwarai ma'am does not need any introduction, I would like to take this opportunity and I feel honored to introduce our today's eminent speaker. Professor Sandhya Vishweshwarai is the chairperson of the Department of Molecular Reproduction Development and Genetics and a co-chair of the Center for Biosystem Science and Engineering, Indian Institute of Science, Bangalore. She also holds the position of an adjunct professor, Faculty of Medicine and Dentistry, University of Bergen, Norway. Professor Vishweshwarai obtained her bachelor's degree in botany from Osmania University, Hyderabad, following which she joined IIT Kanpur to pursue her master's in chemistry. She has done her PhD in Department of Biochemistry, IISc. Later, she worked at Astra Research, Bangalore as a postdoctoral fellow, and then she is a scientist. She joined academics and took up a position of an assistant professor at the Department of Molecular Reproduction, Development and Genetics, IIAC, where she continues to serve as a professor and a chairperson. She is a recipient of numerous fellowships, such as Fellow of World Academy of Science, Indian National Science Academy, Academy of Science, J.C. Bose Fellow, the Fulbright Nehru Senior Research Fellowship, etc. She spent precious time at the lab of Dr. Sabin Earth, Weill Cornell Medical College, New York City, US. Short-term association from the Department of Biotechnology, Government of India for a 3 months visit to University of Missouri, Columbia and Royal Society International Joint Project with Director Roger Buxton, National Institute of Medical Research, UK from 2008 to 11. She was awarded with the Bill and Melinda Gates and Grants Challenges Exploration Grant for her proposal, a small animal model of ETEC mediator diarrhea. Her initiatives to improve translation research in India earned her a Margdarshi Fellowship from the Wellcome Trust or DBT India Alliance in 2017. Professor has been awarded with many prestigious awards, namely YT Tathachari Award 2009, Central Drug Research Institute Award for Excellence in Drug Research 2005, Human Frontier Science Program Short-Term Fellowship to UC Berkeley in the laboratory of Professor John Kurian, Indian National Science Academy Medal for Young Scientists. In addition to her research work, she constantly strived to remove roadblocks for women in science in India. In one notable step, she worked to modify the rules at IISc to extend the tenure assessment period for women on the faculty by 1 year per child. I feel truly privileged to invite Professor Sandhya S. Vishweshwarai ma'am to deliver her talk and enlighten us with her knowledge. Ma'am, we would be honored to know about gut reaction and gut instincts, the underlying biology, and your research work experiences. Please, ma'am.
Thank you very much, Dwani, and it is indeed a pleasure to be invited to give this talk.
Excuse me, ma'am. I would also invite our most honorable Professor D. N. Patel, sir, our Provost of Parul University. He is there. Respected sir, I would like to invite you and I would invite you for a few words. Please, sir.
Ji, Namaskar. In the series of webinar, especially from Applied Science Department, Professor Dhani has arranged so many very good webinar, and I am the man of technology, even though learning a lot from this type of thing. While my study of school, I learned the biology. Then after there was nothing in my touch with the biology, except that I was the Vice Chancellor of Gujarat University. We have a big department of biology, microbiology, and so many others. But as so far this pandemic is concerned and this online teaching learning has started, we consider that whatever this difficulty, challenges to reach into an opportunity. And that's why it is visible that we are getting the speakers like Madam, that they are going to talk with our student and our student will get benefited to that. As so far these speakers are concerned, across the country, across the globe, we are able to manage due to this pandemic. So we sometimes thank the corona that this situation has created. Otherwise, to call someone physically may not be convenient to them and they may not be coming. But very good learning is taking place through this type of activity. And as Parul University is concerned, every day we are arranging such about 8-10 webinars across the various departments, as we have 21 faculty. So each and every faculty, at least alternate day, there is a webinar. And the people like Madam, we are getting really delighted that they are well experienced and very specialized. And our students are the most beneficiary for that. So my best wishes to all students, those who are going to learn from her, and my welcome to her that she has consented to be the part of this type of activity and support the teaching learning activity of Parul University. So Madam, welcome to Parul University, and hope so that our all student stakeholders are the most beneficiary of your talks. Thank you very much. Over to Dhwani.
Thank you so much, sir. Actually, Madam Sir was about to come initially, but he was busy in one meeting. But after knowing about you, our Provost Sir was very delighted to come and wanted to speak with you personally, and he wanted to welcome you. So, thank you, Provost Sir. Thank you so much. We are highly enlightened with your words. Thank you so much, sir. And I would request to Madam kindly start our lecture. Thank you so much, ma'am.
All right, fine. You know, it's really wonderful that universities like yours are taking this kind of initiative, and I kind of agree with what the provost said, that somehow COVID has allowed us to explore different ways of communication and sharing knowledge, which I guess earlier we had never thought was possible. But I do realize now that this kind of webinar and this kind of outreach actually reaches a far larger number of students than just a lecture given in a hall to about 100 or 200 students. The only unfortunate thing is that I'm not able to see you all, and all I can see is a screen in front of me and my slides, which makes it a little bit impersonal for me, but I'll continue anyway. Now, I understand from Dhwani that the audience of students is quite varied. We have students right from the undergraduate stage all the way up to PhD students. And therefore, I thought that I should not really talk too much about the science that I do in the lab, because then it becomes very technical, and I might lose a number of the younger students who haven't yet learned some of the methods that we use in the laboratory. And therefore, I thought that I'd give a general talk about an organ in our body that, perhaps we take for granted until it starts creating problems. And that is the gut. And I'm going to try to convince you today that while many young people are fascinated by understanding the way the brain functions, actually the gut is the organ that controls almost every single aspect of our physiology and biology. And it was Hippocrates who said many, many years ago that all disease begins in the gut. And he realized at that time that any malfunctioning of the gut led to severe consequences in terms of overall organismal biology. So let's get started with the talk. Now, this cover slide, the picture below you shows you a section of the gut, which has been stained with suitable colors just to make it visible. And you can immediately see how complex it is. And I'll talk to you a little bit about the cellular architecture of the gut as we go along. Now, the human gut, this is something that one would have learned with at school. I'd learned about at school. I'm not sure if you can see when I'm pointing. Let me see if I can get the pointer on. Yeah. Can you see the pointer? I hope so. So
Yes,
this
ma'am.
is the esophagus that leads into the stomach. And when I'm talking about the gut today, I will avoid talking about the liver and the pancreas, which are, of course, obviously very important in terms of gut function. But I will focus on this very long convoluted tube, which we know is the small intestine, which ultimately leads into the large intestine and the anus. Now, this gut is obviously extremely important for functions such as food absorption, water absorption, and it's also important to realize that because the gut is exposed to stuff that we take in orally, it has to be able to withstand a lot of foreign objects that enter it and without inducing an immune response. But one of the most characteristic features of the gut, which really honestly was appreciated perhaps a decade ago, is how heavily populated the entire gut is with bacteria. Now, in this section, I'm showing you an electron micrograph of an intestinal cell, which is colored in light brown, and closely adherent to this intestinal cell are bacteria, which are colored green. So you can see that in our gut, we have millions and trillions of bacteria that colonize the gut and have a very important role to play. And it's been estimated that we have almost 1 trillion microbes in our gut. Now, when I talk about microbes, I'm not just referring to bacteria, I'm also referring to fungus and viruses. We don't even know that they exist. But what they're doing is living in very good symbiosis with us in terms of maintaining overall gut health and physiology. The microbes are in such large numbers that they've even been called the forgotten organ. In other words, we're not even aware that these bacteria colonize the gut to such high numbers. These organisms can be called the gut flora, and they live in the digestive tract of every single animal on this planet. The human body, as we have it, consists of about 50 trillion cells. Okay, so I'm talking about liver cells, brain cells, blood cells, etc., etc. But if you were able to count the microorganisms that are found in the intestine, you have almost 500 trillion cells. That is, there are 10 times as many microorganisms in the intestine as there are total cells in the human body. Now, these bacteria don't just sit around doing nothing. They perform very profound metabolic activities. And it's this complex metabolic activity that these bacteria and other organisms do in the gut that resemble that of an organ, such as the liver, for example. And so there are many scientists who believe that the gut bacteria are called a forgotten organ. We're not even aware that such a complex biology exists within our gut. Now, another term for the gut flora is what's called the gut microbiome. And this is a word that you would have seen in newspapers, you would have seen in popular articles, because people are beginning to realize more and more how important the gut microbiome is in physiology. As I said, if you now think of one bacteria, and if you consider all the 500 trillion bacteria there are, you actually have almost 100 times as many genes in these microorganisms as there are in the human genome. We estimate that the human genome has about 20 to 30,000 functional genes, but the total number of genes contributed by the microbes that live in our gut, can be almost 100 times as much. Now, as I said, a number of different types of microbes live in our gut, including fungi, viruses, even unicellular microorganisms. But bacteria make up almost 60% of the dry mass of feces and are present in very large numbers in the colon. It is estimated that there are hundreds, if not thousands, of different species that live in the gut. And what is extremely important for us to realize is just like every human being on this planet has a different fingerprint, every human being has a different microbiome. And this is because of the variety of diet that we consume, our internal physiology that allows certain microbes to thrive and certain not to. So you can even distinguish an individual by analyzing their microbiome at some level. Now, if you can focus on the left-hand side, you will see the different regions of the human gut and the numbers or the varieties of different bacteria that are present in the gut. You have low numbers of bacteria in the stomach, and this is, you could expect, because of the very harsh acidic environment that the stomach has. But as you progress down the gut, the number of bacteria that you find are much, much larger. And you can reach 10 to the power of 9 to 10 to the power of 12 bacteria in the colon. Excuse me. Listed are the kinds of bacteria that one comes across. The major species are Bacteroidetes, Proteobacteria, Firmicutes, and others. Again, very beautiful pictures on the right-hand side showing you the different types of bacteria that can colonize the gut. Lactobacillus is considered a very favorable bacteria. Now, many of you would have heard of what are called probiotics. These are things that are sold now across the counter, which people ask you to take so that you can improve the health of your gut. And the majority of those probiotics contain lactobacillus. And lactobacillus is the one that helps us digest milk products, and it's considered a good bacterium. On the other side are what are called segmented filamentous bacteria. These are these long green things that you see projecting out of the intestinal cell. Now, these bacteria are actually considered harmful because they overactivate the immune system. And so it's this interplay of good and bad bacteria that make a difference in terms of overall gut health. Now, coming back to the different types of bacteria that you see, as I mentioned, in the stomach, the number of bacteria range from about 10 to about 1,000 colony-forming units per ml. But as you move down the gut, you end up at the colon, where you can have almost 10 to the power 11 to 10 to the power 12 CFU per ml. Now, where do these bacteria come from? A child within the mother's womb has a sterile gut. There are very, very few bacteria that can cross the placenta and enter the fetus. All of this colonization of bacteria happens during the life of the individual after they're born. And I'll talk a little bit about this. On the right-hand side are the different types of bacterial species that are present in the gut. You can see that good bacteria like lactobacillus are present to not such high amounts. So now when you take these probiotics, you can, to a certain extent, increase the amount of lactobacillus present in the gut. And it's bacteroidety species that constitute about 30% of all gut bacteria. People have questioned whether microbes in the gut are actually man's best friend. You can raise animals in laboratories, of course, where you can completely remove all bacteria from their gut. And these are what are called germ-free animals. Now, you can generate them either by the treatment with antibiotics, or soon after birth, the mother and the pups are kept in a completely germ-free environment. This is very, very difficult to do, requires very sophisticated air handling systems, as you can imagine, because in the air, we are completely surrounded by many, many types of bacteria and viruses. Now, if you grow these germ-free rats and you start feeding them perfectly sterile food, you will see that they require almost 30% more calories to maintain the body mass of normal rats. What that means is that the microbes that colonize the gut are extremely important in maintaining body mass in normal organisms. And if you deplete organisms of their microbes, they actually require much, much more calories to be able to sustain and be healthy. It's understood now that the bacteria of the GI tract contribute to the nutrition. They do this by liberating simplified carbohydrates, amino acids, and vitamins that can be absorbed by the gut. For example, the food that we eat is made up of complex proteins, complex carbohydrates. They need to be broken down into simple sugars by the various bacteria that line our gut. And it is they that allow the formation of these simple molecules, which can now be taken up by the intestinal cell. Now, a very good example of this is an organism which is called B theta iota micron. And its genome, or the DNA content of this particular bacterium, has a very, very large number of genes that contribute to carbohydrate binding and carbohydrate breakdown. Now, these organisms live in the gut. So this is a question of coevolution, where you have bacteria that realize that they're in an environment where complex sugars come to them free of cost in that sense, because that's what we eat. They're able to latch on to these carbohydrates, and they're able to degrade them into simple sugars, which can now be taken up by the bloodstream. Now, let's talk a little bit about how the microbiome actually establishes itself in humans. Human microbial colonization begins at birth. And there are changes in species abundance till 3 years and beyond. And so you end up finally getting microbiota that are adult-like. Now, over the first 3 years of the life of a child, there is a clear exposure to significant microbes. Now, where do these microbes come from? When a child feeds from its mother, there are bacteria on the mother's skin which are naturally introduced into the gut of the feeding child. Children, as you know, keep on putting their hands in their mouths and their feet in their mouths, and they keep picking up stuff off the floor and stuffing it into their mouths. All that is good to a certain extent, because in doing so, they are now introducing these microbes into their gut, which, as I said, have beneficial effects. When you start crawling, or when children start crawling and they put their hands on floor surfaces, this is another time, because that hand, the same hand, goes into their mouth a second later. And so all of these very natural tendencies of children is the way we have evolved to be able to colonize the gut by microbes in our environment. Now, as you all know, children also suffer more infectious diseases than adults because they do touch stuff that is not good for the system. But in order to avoid that, this does not mean that we continuously keep washing the hands of our children and prevent them from putting things in their mouth, etc., etc. And actually, this modern changes in lifestyle, which one has seen in the Western world dramatically, where you have improved sanitization, where more and more women are having cesarean sections to give birth. There's an overuse of antibiotic usage. There's a lot of immunizations. All of these things can shift the microbiota. And it has been argued that it's all these human interferences that are actually the potential drivers of the sudden increase in immune-mediated disease in the developed world. Now, if you think about it, our body is supposed to react to bacteria or foreign organisms that it doesn't recognize. But however, you know very clearly that most of us do not have any reaction to the microbes that are in our gut. In fact, we don't even know that they're there consciously. And this has happened because we have become sensitized to the presence of these microbes, and the body is no longer recognizing them as foreign bacteria. And it's been able to do this during the colonization of the gut that happened from infancy onwards. If you remove these microbes from children by, changing your lifestyle, etc., the body then becomes far more susceptible to the introduction of these bacteria at later stages, which now results in immune-mediated diseases and autoimmunity. So here we are. Here is this child growing up. At birth, there are very, very few bacteria. And the bacteria are actually introduced into a child during its passage through the birth canal of the mother. So there are a number of bacteria that line the birth canal in women. And so as the baby passes out through the birth canal, it picks up some of these bacteria. And that is a perfectly normal and perfectly healthy thing to happen. By the time the child is about 1 month old, it started touching things around it. It started touching its mother's skin, its father's skin, and the number of bacteria then start increasing in the gut. Of course, by about 6 months, when the child is crawling around and putting stuff into its mouth, again, you have a very large variety of microbes that can enter. And this goes on till the child is around 2 to 3 years old. But notice how the composition of the bacteria that are predominant in the gut of these children changes over the course of their growing old. And children achieve an adult microbiome by the time they're about 5 to 6 years old, because it's around that time that they've started eating the normal food that an adult is likely to take. You can see that bacterial diversity increases as the child grows older, and inter-individual variability starts decreasing. However, there is still a very unique microbiome that each of these children has. Now, what are the factors that influence the gut microbiome? As I said, the first is prenatal exposure to amniotic fluid. Now, there are a few bacteria that do come into the amniotic fluid. All of you know that this is the fluid that surrounds the developing fetus in the mother's womb. And some of those bacteria are swallowed by the developing fetus and can colonize the gut. The mode of delivery, as I mentioned, you have cesarean sections. Now, what happens then is the child is lifted out of the womb without having passed through the birth canal. And what is being noticed is that children born of cesarean sections actually have a higher risk of the development of inflammatory bowel disease, obesity, and celiac disease. Now, this is an increase in incidence. It doesn't mean that every child born of a cesarean section is going to develop these disorders. But it's interesting to note that when a child is born without having been exposed to the bacteria that are present in the birth canal of the mother, there can be long-term consequences in terms of overall health of that individual. Now, let's take the child after it's born. Breastfeeding and formula feeding. When a child actually feeds from the mother, it ingests between 10 to the power of 5 and 10 to the power of commensal bacteria every day. However, when you prepare formula feed for a child, you boil the water, you sterilize everything, you sterilize the bottle, and the child is no longer exposed to all these normal, healthy bacteria that are found in human breast milk. So again, this is a lifestyle change, and therefore, for very many reasons, breastfeeding is the best way to take care of a young child. Now your child is introduced to solid foods. At this time, there is definitely a shift in the diet. From predominantly a milk-based diet, you now start providing the child complex proteins, complex carbohydrates, and therefore the kind of bacteria that are present in the gut need to change in order to be able to cater for this altered diet. Now, what about geography? Obviously, as you all know, the diets of humans around the world vastly differs. The Indian diet is extremely different from a diet of a normal American, for example, or a British individual. Similarly, the South American diet is found to be a little bit more similar to us because of the use of spices, etc. So there are huge differences in the kind of diets that are seen in populations around the world. We also know that populations around the world have slightly different genetics. They look different, they talk different. And it's all this whole combination of just not the genetic background of the individual, but also regional diets, cultural practices, for example. All of these things influence the kind of gut microbiome present in an individual. And finally, of course, humankind has discovered antibiotics. There are broad spectrum antibiotics that target many, many different types of bacteria. And many of them are prescribed somewhat indiscriminately to young children to protect them from disease. But this again affects the composition of the microbiome, which can in fact have deleterious consequences later. So now, many, many years ago, Charles Darwin, in his book, The Expression of the Emotions in Man and Animals, made this very profound statement. At that time, of course, nobody knew anything about the microbiome. People were only just about beginning to understand the kind of anatomy of the gastrointestinal tract. But he said, the manner in which the secretions of the alimentary canal and of certain other organs are affected by strong emotions is another excellent instance of the direct action of the sensorium of these organisms, independently of the will of any serviceable associated habit. Now, this is very, very complex English, but what essentially he was saying is that when your mood is different, or when you're thinking something different, you're able to sense changes in the way your gut reacts, and vice versa. For example, if you have an exam ahead of you, you tend to get a little nervous. And when you tend to get a little nervous, some of us start experiencing a little bit of diarrhea. They experience a little queasiness in the stomach. We all know that, we all have that phrase, right? My stomach is not feeling very good because I've got an exam ahead of me. So this is a way by which you can clearly notice that something that happens in the gut affects the mood and vice versa. And this crosstalk between the gut and the brain is what is called the gut-brain axis. And this is the biochemical signaling that takes place between the gastrointestinal tract and the nervous system. Now, people knew that it existed for some years, but it's only in the last very few years that we now realize that this interaction is actually driven by the functions of the intestinal microbiota. How does this happen? So here is the epithelium of the gut, and you have a number of bacteria that are living in the intestinal lumen. These bacteria contain genes in them that actually make neurotransmitters. Now, these neurotransmitters are sent into the intestine, where they now get into the blood vessel, and then they move to the brain and affect things like mood cognition and emotion. In an opposite direction, the hypothalamus produces various hormones, which now affect cortisol levels. This affects the movement of muscles that line the gut. And this again now starts affecting the bacteria that are present in the gut. These bacteria are recognized by immune cells that are below the epithelium of the gut. Now, these immune cells will again now maybe destroy these bacteria or allow them to survive. Bacteria produce molecules which are called short-chain fatty acids. These are lipids, which again enter the intestine and enter the blood vessel and again move all the way to the brain. So it is this very complex interplay between the brain, the gut, and the bacteria or the microorganisms that live in the gut, which constitutes the gut-brain axis. And so there are very, very nice reviews where you can talk of what are called mind-altering microorganisms. That is the impact of the gut microbiota on the brain and behavior. Let's talk about this a little bit more. Let's talk about Parkinson's disease. Now, all of you would have heard of Parkinson's disease. This is a disease where you lose control of the way your muscles move. You find it difficult to walk. There are slightly jerky movements. And it's been known that this Parkinson's disease is caused by or misfolding of a protein called alpha-synuclein. Now, E. coli that lives in the gut makes a protein which is called curly. Now, this protein curly can prompt other proteins, including alpha-synuclein, to misfold. And so some researchers suspect that these misfolded proteins, these curly proteins, enter the nerves that line the gut, make their way to the brain, and then trigger the misfolding of synuclein, which now leads to Parkinson's. So this is one example which scientists have identified so far. Now, there's another bacterium, which is called Akamancia Mucinephila. And this bacterium has been found to be associated with motor neuron disease. Now, many of you would have heard of motor neuron disease. This is what Stephen Hawking had, where you lose control of your muscles and you lose control of movement, and you ultimately end up being paralyzed. It's been found that this particular species of bacteria makes a molecule which is called nicotinamide. Now, nicotinamide is also known as vitamin B3. The nicotinamide produced by this bacteria travels in the bloodstream up to the brain and can actually improve the symptoms of motor neuron disease, at least in mouse models. What does this mean? There is now, in patients with motor neuron disease, if you're able to colonize their gut with this particular bacteria, would it be able to help them and alleviate some of their symptoms? Now let's talk about segmented filamentous bacteria. I'd shown you a picture of them a little earlier. These bacteria sit in the gut, and when you have infection, bacteria stimulate a certain type of immune cells called T helper 17 cells. These cells now produce cytokines. These cytokines, such as IL-17, can pass into the placenta and actually reach the fetus of the brain. And what has been shown is that in mice, an infection in pregnancy can set off this cascade of activity. And this production of immune molecules, when it reaches the fetus, can actually provoke something like autism spectrum disorder or autism-like behavior. Now, this is science that's been discovered perhaps in the last couple of years. And I'm sure that there are going to be many, many more examples where we understand how gut microbes actually can cause very long-term effects in neurological function. And things like autism spectrum disorder, etc., are stuff that would obviously require some intervention during pregnancy if this link is indeed found to be true in humans as well. So now let's get down to the anatomy of the intestinal cell a little bit and how the intestine actually responds to the environment. This is a pseudo-colored image of the way the intestine looks in the small intestine. You have these large finger-like projections, which are called villi, which project into the lumen. Now you have so many villi, and they are covered even by other small microvilli projecting off the surface. And this increases the absorptive capacity of the gut so that it's able to take up all the nutrition and all the food that you eat during the day. Now, this photomicrograph shows you a certain pathogenic strain of E. coli that has colonized the gut. And you can see that what it's been able to do is completely destroy the microvilli that were projecting. This is the surface of the epithelial cell. And you can see that this organism has completely destroyed all these microvilli. One very interesting fact that many of you may not know is that the gut is one of the most proliferative tissues in our body. And what happens is that every 4 days, we generate the complete epithelial cell lining that coats the gut. There are cells at the bottom of the gut, which are called stem cells. These are intestinal stem cells. They continuously divide, and as they divide, they push up these cells to the tip of the villus. At the tip of the villus, these cells undergo a process of cell death called apoptosis, and the cells slough off into the lumen of the gut. But they are continuously replenished by these new cells that grow from the crypt. So you have a region of the gut where you have proliferation. There's a step of differentiation. I won't go into too much detail here, but there are a variety of different cell types in the gut. And then finally, at the top, you have a process of apoptosis. Now, since this tissue is so rapidly proliferating, it means that there's obviously exquisite control as to its rate of proliferation. Because as we all know, uncontrolled proliferation in cells leads to tumorigenesis. And instances of colorectal cancer or gut cancer are really quite rare in comparison to other more common forms of cancer, such as oral cancer or breast cancer, etc. So now, what do these cells do and how do they communicate with their environment and the microbe? Now, this is something for process which is called signal transduction. Conversation or interactions between living organisms is absolutely natural. And even though we're not aware of it, at the level of a single cell, a huge amount of communication is occurring. And this is much, much more than all the billions and billions of cell phones that we might have on the planet. And signal transduction is the study of how a cell communicates. And it can be argued that if a cell, a living cell, cannot receive or respond to signals from the environment, it could not adapt to its behavior over time and would soon be outcompeted by those that could communicate. Now, I cannot go into this huge area of signal transduction, which is really the focus of research in the laboratory. But I'll talk to you about two important molecules that are important, at least as far as the gut physiology is concerned. And these are small molecules called cyclic nucleotides. Cyclic AMP and cyclic GMP, which is synthesized in the body from ATP and GTP, respectively. Now, these small molecules, which are called second messengers, and I'll tell you why a little later, were discovered by Earl Sutherland in 1971, for which he received the Nobel Prize. And the citation for the Nobel Prize said that when you discovered cyclic AMP, you discovered one of the fundamental principles involved in the regulation of essentially all life processes. And that is absolutely true and has stayed true even with the advances we have in today's understanding of biology. Now, cyclic GMP was always considered the poorer cousin of cyclic AMP. However, it became famous when it was identified that nitric oxide, with a gas, regulates cyclic GMP levels in the cell. A number of other molecules also can modulate cyclic GMP in the cell. These are peptides called atrial natriuretic peptides, heat-stable toxins, etc. And it was the discovery of nitric oxide and its link with cyclic GMP that again resulted in the award of a Nobel Prize to these three gentlemen, Birchcott, Ignarro, and Murad. So you have a recognition of the importance of these second messengers in basic physiology, and importantly, in intestinal functioning. Now I'll come to why these second messengers are so important in intestinal function. In order for the food to pass down the gastrointestinal tract, there has to be some amount of lubrication. And this lubrication is provided by water that is secreted by the intestinal cell, and also by the number of ions that are going out of the intestinal cell or being absorbed by the intestinal cell into the blood vessel. And you have a number of toxins produced by bacteria that colonize the gut that cause diarrhea. Many of you would have heard of a molecule called the Cystic Fibrosis Transmembrane Conductance Regulator, or CFTR. This is a chloride channel which is found in the membrane of intestinal cells, which secretes chloride ions. There are patients who have mutations in this particular protein, and those individuals have a disease called cystic fibrosis. Now, what happens is that chloride ion secretion by CFTR is blocked now in these individuals. And because chloride ions are not secreted by the gut, this also affects the secretion of water by the gut. And therefore, the inner lining of the gut and any other epithelia which harbor cystic fibrosis becomes very mucus-like and very thick. And this thickening of the mucus allows a greater number of bacteria to colonize this mucus. And this is actually what is seen in the lungs of cystic fibrosis patients, where they have a collection of a very large number of bacteria as a consequence of misfunctioning CFTR. And those bacteria are the ones that cause complications such as pneumonia, etc. Now, bacteria live in the gut, and many of these bacteria which are pathogenic or which cause disease produce enterotoxins that regulate intestinal cell function. We've all heard of Vibrio cholera. This is the organism or the bacterium that causes cholera. And it produces a toxin called cholera toxin. And I'll show you in a little while how that affects intestinal epithelial cell functioning. There are other strains of E. coli which produce another type of toxin called the labile toxin and the heat-stable enterotoxin. And Shigella species produce Shiga toxin and Shiga-like toxin. And these are one of the major causes of diarrhea that all of us may have experienced over the course of our lives. Now, these bacteria are very smart. What they do is utilize the normal machinery that is found in the human intestine to their advantage. So let's look and see what cholera toxin does. Cholera toxin, this is Vibrio cholerae, produces this cholera toxin, bit of a complex protein molecule, which now enters cells and interferes with the production of cyclic AMP. Cyclic AMP levels are increased very markedly in the intestinal cell. This results in a signal transduction event, which ultimately results in modification of the cystic fibrosis transmembrane conductance regulator by phosphorylation. What happens then is that Cftr starts secreting too much chloride ions into the intestinal lumen. Now, in order to maintain osmotic neutrality, the intestine starts secreting a lot of water. And this is the diarrhea that you experience, a large amount of water secretion and a large amount of chloride secretion. Now, as you know, cholera in many cases can be fatal. And that is because people infected with Vibrio cholerae can secrete almost as much as 5 to 10 liters of water every day. And therefore, their fluid replenishment needs to be very, very extensive. Otherwise, they will die of dehydration. So this is a mechanism by which a bacterium toxin actually affects and interferes with the functioning of the normal intestinal cell. So now I think I've given you an overview of what the intestinal flora does. As I've said, in many cases, they're protective. They can many times compete for pathogens. So if you have a good microbiome in your gut, you can actually fight off pathogens which produce these toxins and therefore remain healthy. It can take care of nutrient competition, and many of these bacteria, for example, produce antimicrobial factors such as bacteriocins and lactic acid that can actually ward off other pathogenic bacteria. And these are what are called the commensal bacteria. In other words, the ones that remain are what are called commensal bacteria. Intestinal flora also have structural functions. For example, they serve as a very good barrier so that foreign objects don't enter the intestine and in the body that easily. They can induce the production of a molecule called IgA, which is an immunoglobulin, which helps in protecting against bad bacteria. It helps in maintaining what are called tight junctions between the intestinal epithelial cell so that all sorts of unnecessary stuff is not absorbed by the intestine or reaches the blood vessels below the gut. And it's a only regulated uptake of specific molecules that the intestine is able to do. In terms of metabolic function, I think I touched upon that. They can produce many of the vitamins. These bacteria can produce many vitamins that are absolutely essential for our well-being, such as vitamin K, biotin, and folate. Our bodies do not make these vitamins, so they need to come from the microbes that live in our gut and, of course, from our diet. Many of these bacteria control intestinal epithelial cell differentiation and proliferation. This is very advanced biology that's happening in many groups, including mine in the laboratory. Many of these bacteria can actually metabolize dietary carcinogens. So our diets these days sometimes contain stuff that's really not good, but these bugs can take care of them, break them down, and get rid of them from our body. And as I said, many of them can synthesize bacteria, vitamins. Now, something that's been an obsession perhaps in the health sphere over the last couple of years has been the SARS-CoV-2. And I thought that before I ended, I would just tell you a little bit about how SARS-CoV-2 interacts or links with the gut. You know, it's been found that almost 50% of COVID patients have GI symptoms, that is gastrointestinal symptoms. This can range from losing a sense of smell and taste to very frequent diarrheal episodes. And many of these GI symptoms can continue even after COVID patients recover. The virus can be detected in the feces in about 50% of infected subjects. Now, what that means is somehow, even though we have inhaled the virus into our lung, it's able to reach the gut to some extent. Now, this virus, as we all know, and I'm sure many of you would know, uses a protein called ACE2, which is its receptor on the host cell. So SARS-CoV-2 binds to this receptor and enters the host cell. And this receptor ACE2 is actually expressed at high levels in the small intestine, especially in the types of cells that I showed you, the proximal and what are called distal enterocytes. Now, the entry of SARS-CoV-2, while it can bind to ACE2, also requires a protease, that's an enzyme that cuts a particular peptide bond, called TMPRSS2. And TMPRSS2 is also highly expressed in the ileum and the colon. What this means is that this virus can actually invade enterocytes and the digestive tract. And research that has happened or occurred in the last couple of years suggests that the kind of microbiome that you have in your gut determines the outcome of COVID infection. If you have a good microbiome, you have what are called eubiosis, but this good microbiome is dependent on age. So for example, we know that as you get older, the microbiome starts changing, becomes less healthy in inverted commas. Things like comorbidity. We all know that individuals with diabetes or blood pressure seem to fare far more poorly when COVID infections come in. Those kinds of chronic diseases can affect the microbiome. All of these things now result in a lower effect of COVID in terms of disease. Probiotics, prebiotics, good fruit and vegetable consumption allow you to experience COVID either asymptomatically, that is, you don't even know you have the virus, or you are symptomatic but to very moderate levels. Now, what happens in the gut is because of changes in the microbiome, resulting in altered fatty acid production, production of inflammation mediators, etc., results in what is called dysbiosis, or not a very good microbiome in your gut. As a result, you end up with all of these complications, such as hyperinflammation, comorbidities, etc. And of course, if you're administered antibiotics, again, the gut microbiome will change. And it's all of these factors that people have now linked to severe to fatal COVID-19. So as you can see, the gut is integral to the physiology and well-being of our organism. And we're appreciating more and more how important the microbiome is in being able to regulate the health and well-being of an individual. Disruptions in the gut microbiome, antibiotic usage, diarrhea can lead to temporary malfunction. And natural processes, such as diet, replenish our gut microbiota. And in fact, the traditional Indian diet and the traditional Indian lifestyle is actually, in a way, very healthy and good for our gut. Thank you very much. And I would be pleased to take questions.
Thank you so much, ma'am. We are really fortunate to hear you in person. I'm sure all the viewers and are motivated and encouraged and get a lot of from your descriptive and illustrative presentation. Thank you so much, ma'am. I have received one question from a student. Apart from the syllabus, what you have taught, that ma'am, we would like to know about your research experience while you working primarily in the area of signal transduction, cell biology, and protein structure.
I'm not very clear what the student wants from me at that time. As I said, I wanted to avoid talking about my research directly, but one of the most exciting things that one does in a research laboratory is actually interact with students, because students come in with a fresh perspective, a fresh way of looking at things. In terms of signal transduction, I focus on basically signaling through cyclic AMP and cyclic GMP. The interest lies in bacterial toxins that cause diarrhea. We have identified the receptor for this particular toxin. We were also able to show that mutations in this receptor cause a kind of rare diarrheal disease in humans, which is seen from children onwards, from infants onwards. We have now developed mouse models which try to mimic that human disease so that we can understand a little bit more about how this whole receptor signals in the gastrointestinal tract. But this receptor has roles in terms of cell proliferation. Therefore, if you have mice which have this receptor knocked out of their genome, what are called knockout mice, you will see that their gut is more proliferative and they're more susceptible to getting colonic tumors. So this is the gist of what we do in the lab. But I mean, as I said, I didn't talk about the research in this particular talk.
Okay, thank you so much, ma'am. There is one more question. Can you refer any special book for the study of gut-brain axis?
Yeah, so, this is research that is happening as we speak. And so while you might come across a couple of reviews in scientific literature, which is where I got my information from, there is no book yet because there isn't enough knowledge to be able to write a simple textbook of this. So you will find these reviews in many leading journals such as Nature Reviews, etc. If you do a Google search, you'll find them. They're not easy to understand because they're written for the scientific audience, but there are very, very good books available on the microbiome. And in that, you will find perhaps a chapter where they talk about the microbiome and the gut-brain axis. But if you're not interested in looking at links with the microbiome, the way the gut talks to the brain are papers that are published in Nature last week or the week before. So this is really very, very current stuff, and you'll not find it as a book or a textbook that somebody can read.
Thank you so much, ma'am. One more question. We would like to know your views on the role of a woman in the field of science since past decades.
Why only science? Women are essential for every single activity in this world. And in many ways,
Yeah.
I think women are more essential than men because we give birth, right? Which
Yeah,
so far,
absolutely.
it's not been easy for men to do. So, as far as science is concerned, there are certain disciplines of science where you don't see many women. For example, in the areas of physics, mathematics, engineering sciences. All that must change. And I think it all depends in societal's expectations of what women are supposed to do. You'll find a lot of girls who take up biology. For example, in our institute, I would say that more than 50% of our PhD students are women. But unfortunately, what happens is that they drift out of the system later. And if you look at the number of women professors in the institute, it's less than 10%. So somewhere along the way, there is what is called a leaky pipeline. And, women must now believe that they are able to do whatever their male colleagues can do, maybe better than their male colleagues in many ways, because women are generally more empathetic. They're a little bit more sympathetic to others, and they're less aggressive and violent in the way they deal with situations. But it's up to the girls to really believe that they can, and there is nothing that can stop them. They just need to believe in themselves. And of course, unfortunately, parents need to believe in them as well.
Yeah.
Otherwise, fortunately, in countries like India, I sometimes get the feeling that I need to talk to parents rather than talk to girls, because I think the girls believe they can do it. But the parents feel that, oh, well, she needs to get married, she needs to settle down, she needs to do this, she needs to do that. And, that's where the problem is. But once you get into the scientific field, for example, I tell all my students in my lab that they're as equal as anybody else in the lab right now. They're getting the same salary. They're getting the same opportunities as their male colleagues. Make use of them. Don't step back and don't say that, oh, what will my parents say, or what will this happen? Why do you need to say that? You're financially independent now. You decide to do what you want to do. So the
Exactly.
minute you achieve financial independence, I think you need to stay strong.
Thank you so much, ma'am. And there is one more question. Which advice would you like to give our young students who are research enthusiastic and pursuing their UG and PG programs in biosciences?
Yeah. I mean, I think there are a number of opportunities available for students now who are interested in the biological sciences. Keep looking out for the KVPY program. There are Inspire fellowships. Getting admission into good institutions in India is difficult. And this is just because we have such a large population, right? So, I mean, there are so many people, but not enough opportunities that make that, getting into a place like IASC or NCBS or TIFR is not easy. But I'm really of the belief that if you have the motivation and the drive, when you come for interview, we will be able to recognize that. And we will select you from whichever university you are from. You don't have to be from Delhi or one of these fancy places to get in. There are a large number of opportunities. You just have to look for them. Nothing is going to be given to you on a platter anymore. That is what internet has done, right? You have to just search. You have to spend time, figure out what's available, and make sure that you take up these opportunities when you do.
Yes. Thank you. Thank you so much, ma'am, for sharing your deep knowledge and edifying words of wisdom with us. I'm
It's
sure
a
it has
pleasure.
elevated and uplifted all the viewers to accomplish their goals with the same zeal and zest. So I'm very deeply thankful to you. I would like to request our provost sir, Dr. Emin Patel sir. Uh, hello, sir. I think yes, sir. Sir, we like to request you to kindly have a few words on this occasion. Please, sir.
So I heard the entire talk and just brush up my knowledge as far as this biology and that area is concerned. And your question also I was hearing regarding that the role of women, especially in the research and so. So I can talk on that. Recently our Olympic is going on. You see that the contribution of women is more than the men's. As medals are
In terms of
secured more
medals.
by the women than the men.
Yeah,
And the previous all records we have seen that the more medals are captured by the women. So the role of women is now everywhere. As so far our teaching community fraternity is concerned, there is no question of reservation of 50% as government has temporarily kept it that 50% women to be there. But here we see that they are more than that. And as by the virtue of that habit and everything, they are more successful as so far the research and teaching learning is concerned. So we should appreciate that the contribution of women in the society. And obviously, all such a role model women are also be there. They have did very fantastic work as so far the welfare of society. And naturally, now there is no barrier between men and women. All are equal. And we hope so that we will work in that way only. No doubt
Thank you.
our society is conservative and thinking little bit that they are the supreme compared to the women. But that is not now, after a lot of
sure.
education, I think that also get vanished. So my best wishes to all the students, those who are beneficiary from the talk of the Sanjay ji, and hope so that in future also, whenever our Madam Dhani may call you, you please contribute to us and our student get benefit.
Sure.
Thank
I
you
will
very
do
much.
that.
Over to you, Dhani.
Thank you very
Thank
much.
Thank you so much, ma'am. I would really like to thank you. Thank you so much, sir. You always help us and you always inspire us to do all the different kind of activities. Always we get a platform on Parul University to share our contribution. And ma'am, you are a dream life of my personal and also for many girls, those who are listening you today, you would be able to find it out that there are multiple mails and questions I'm having right now, but due to the constraint of time, I will not be able to share it with you. But definitely, I would like to thank you from these girls also and the different students, those who are
Sure.
listening you. Thank you
Thank
so
you very
much,
much,
ma'am.
Dhwani. Thank you so much. Thank you very much. It was a pleasure. It was a
Thank you
pleasure.
so much, ma'am. Thank you. I would like to pay thanks to all the viewers also. Again to Professor Amin Patel sir and Dr. Girish Babu K sir who is HOD. And thank you so much everyone. Thanks a lot.
Thank you, Dhwani. Bye bye.
Thank you.
Bye bye.
Thank you, ma'am.